In this trial, we studied the safety and efficacy of combination of a PI3K inhibitor called Parsaclisib in combination with standard immunochemotherapy, including R-CHOP or polar R-CHOP in patients with newly diagnosed diffuse large B-cell lymphoma with high-risk features. So we know that we have been always making efforts to improve upon R-CHOP to improve the outcomes of patients with newly diagnosed DLBCL over the last 2 decades. There are many newer agents that have been combined with R-CHOP in randomized trials, but unfortunately we have not made much progress except for the recent Polarix trial, which used a similar approach where they used polatuzumab vedotin in combination with R-CHOP, and they showed a modest progression-free survival benefit compared to the older standard R-CHOP and made it another standard of care option. So going back to the drawing board, we were studying which other pathways can we target in frontline large cell lymphoma. And we learned that the PI3K and mTOR pathway is important in both the GCP subtype and ABC subtype of large cell lymphoma. Our group actually has done a previous trial studying the combination of everolimus, which is an mTOR inhibitor, in combination with R-CHOP, which showed a very good safety data and a very encouraging efficacy signal, where 23 of the 24 patients achieved complete remission and remained in disease-free status at 2 years, which is an important landmark for large cell lymphoma. In this trial, we're shifting our attention to one step up to the PI3K kinase, and we chose a selective and efficacious PI3K inhibitor called Parsaclisib, and combine that with immunochemotherapy. So we had 3 parts of this study: phase 1 dose finding, phase 1b dose expansion, and then finally added a small cohort of safety leading, looking at the combination of Parsaclisib and polar R-CHOP. So in the phase 1 part, we tested 2 dose levels. We didn't see any dose-limiting toxicity. And we have determined that the Parsaclisib 20 milligram daily for 14 days out of the every 3-week cycle of R-CHOP is the dose to be further tested.
Okay.
And then we did a phase 1b dose expansion, looking at the efficacy. In the polar safety leading cohort, we took the MTD of the Parsaclisib, which is 20 milligram daily for 2 weeks again, and studied the significant toxicity of that and efficacy in a small cohort. So in terms of safety, looking at the entire cohort of the polar safety leading, we saw only 1 of 6 patients had a grade 3 diarrhea, which would be considered a significant toxicity. But otherwise, this regimen is considered safe for further expansion as well. In terms of common toxicity, if we combine all the patients in the trial, most of the high-grade toxicity are hematologic, like neutropenia, lymphopenia, thrombocytopenia, and anemia, which we would see with standard immunochemotherapy anyway. I don't think the PI3K kinase inhibitor added too much to that. And interestingly, of the non-hematological toxicity of interest, such as diarrhea, liver toxicity, skin rash, we saw very few, as 6% of diarrhea, 2% of rash, actually no liver toxicity. That's a high grade. So it's important that when we use PI3K inhibitor in an intermittent fashion and a fixed duration fashion in combination with steroids, it seems like the safety is very good. Now moving on to efficacy, we saw very high response rate in this trial. Overall 98% response rate, the CR rate is 92%. In terms of time to event data, the progression-free survival at 2 years is 85% for both cohorts, and overall survival in our R-CHOP cohort at 2 years is 95%. And the polar R-CHOP small cohort, every patient was alive at the last follow-up. So taking together these data from this phase 1b trial with 49 patients treated, we think the combination is safe. And also we saw very encouraging efficacy signal in terms of response rate, progression-free survival, and overall survival. And we do think these data support further exploration of targeting PI3K mTOR pathway in frontline therapy for large cell lymphoma in the future.